Welcome to Biomindscape. Today we are going to know this chapter Biotechnology Principle and Processes. Biotechnology deals with the technique of using living organism or enzyme from organism to produce and process useful to human. For example, making curd, various type of bread or wine, and which are all microbial medial process. They all are formed by some processing in biotechnology. However, it is used in the restricted sense in today's reference. And to achieve the same genetic modification on a large scale, these techniques are included under biotechnology. For example, in vitro fertilization lead to test tube baby, synthesizing a gene and using it, developing a DNA vaccine or correcting a defective gene, and all part of the biotechnology. Now here in ncert it is given that the European Federation of Biotechnology EFB has given a definition of biotechnology that comprises the both traditional and modern molecular biotechnology and the integration of natural sciences and organisms, cell part thereof, and molecular analogues for products and services. Now let's know the principles of biotechnology. Among the two core techniques, they are both of the modern technology are genetic engineering and bioprocess engineering. So in genetic engineering, it is a technique to alter the genetic material, for example DNA and RNA, and it is introduced in the host organism and thus change in the phenotype, that is physical appearance or of the host organism. And bioprocessing engineering is maintenance of the sterile or microbial contaminated free ambience in chemical engineering process to enable the growth of only a desired microbe eukaryotic cell in a large quantity for a manufacturing of biotechnological product like antibiotics, vaccine, enzyme, etc. Let us know the development of the principle of genetic engineering. We all know that there are some advantages of the sexual reproduction over asexual reproduction. The former provide opportunity for variation and formulation of we can form unique combination of this genetic setup, some which may be beneficial to organism as well as the population. Asexual reproduction preserves the genetic information while sexual reproduction permits the variation. So traditional hybridization procedure used in plant and animal breeding very often lead to inclusion and multiplication of undesirable gene along with the desirable genes. And this technique of genetic engineering, which include creating a recombinant DNA using the genetic clone and gene transfer, and overcoming these limitations and allowing us to isolate and isolates and introduce only a set or a desirable gene without introducing the undesirable gene into the target organism. For example, if we see a fate of DNA which is somehow transferred into an alien organism, most likely these pieces of DNA would not be able to multiply itself in a progeny cell of an organism, but when it get integrated into the genome of the receptor, it may multiply and be inherited along with the host DNA. So this is because the alien piece of the DNA has become part of the chromosome, which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating the replication. For multiplying of any, this piece of DNA is needed to be the part of the chromosome which has a specific sequence, which is also known as the origin of replication. Thus an alien DNA is linked in the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can be done by cloning or making multiple identical copies of any template DNA. So let's now focus on the first instance of construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, autonomous replicating circular extrachromosomal DNA of Salmonella typhi. Stanley, Cohen and Herbert accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid, which was responsible for antibiotic resistance. This cutting of DNA at specific location became possible with the discovery of its so-called molecular scissor, that is the restriction enzyme. The cut piece of DNA was then linked with the plasmid DNA, and this plasmid DNA acts as a vector to transfer the piece of DNA attached to it. You probably know that mosquito act as an insect vector to transfer the malarial parasite into human body. I have given a video on which retrovirus infection and life cycle of the Plasmodium vivax I have done. You can watch that video. So in that also we have to now continue with the linking of this antibiotic resistance gene with the plasmid vector become possible with the enzyme DNA ligase, which act on a cut DNA molecule and joins their end, and this make a new combination of the circular autonomously replicating DNA, creating in vitro, and it is known as recombinant DNA. When this DNA is transferred in the E. coli, a bacteria closely related to Salmonella, it could replicate using a new host DNA polymerase enzyme and can make multiple copies, and this ability of multiple copies of antibiotic resistance genes in E. coli was called cloning of antibiotic resistance gene in E. coli. You can hence in for that there is 3 basic steps in genetic modification, that is identification of the DNA with desirable gene, introduction of the identified DNA into the host, and maintenance of the introduced DNA in the host and transferring of the DNA to its progeny. Now the tools of the recombinant DNA technology, we know that the genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools: the restriction enzyme, polymerase enzyme, ligase, vector, and the host organism. So restriction enzyme, in the year 1963, 2 enzymes responsible for restricting the growth of bacteriophages E. coli were isolated. One of these was added the methyl group to the DNA, while the other cut the DNA. So later was called restriction endonuclease. The first restriction endonuclease, HEN2, was functionally dependent on the specific DNA nucleotide sequence, was isolated and characterized by the 5 years later, and it was found that HEN2 always cut the DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs, and this specific base sequence is known as the recognition sequence for the HEN2. Beside HEN2, today we know more than 900 restriction enzyme has been isolated over 230 strain of bacteria, each of which recognize different recognition sequences, and this conventions of naming this enzyme is the first letter of the name come from the genus and the second 2 letter come from the species of the prokaryotic cell from which they were isolated. Example, EcoR1 comes from the Escherichia coli RY13, and EcoR1 letter R is derived from the name of the strain. Roman number following the name indicates the order in which the enzyme were isolated from the strain of bacteria. So restriction enzyme belong to the large class of enzyme called a nuclease, and these 2 kinds, exonuclease and endonuclease. The exonuclease remove nucleotide from the end of the DNA, whereas endonuclease may cut at a specific position within the DNA. So each restriction endonuclease function by the inspecting sequence, and it was bind to the DNA and cut the each of the 2 strand of the double helix at specific point in the sugar phosphate backbone. So each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. So do you know what is an palindromes are? They are the same word where they read from the both forward and backward. For example, Malayalam, the spelling of Malayalam MALAYALAM. So you can read it from both sides. So as again a word, palindromes were the same words we can read in both direction, and this palindromes in the DNA is a sequence of base pair that read same on the 2 strand where orientation of the reading is kept the same. For example, following sequence read the same on the 2 strand 5' to 3' direction. This is also true for reading 3' to 5' direction. Here in NCERT it is given 5' GAATTC to 3' and in 3' it is CTTAAG 5 dash. So the restriction enzyme cut the strand of the DNA a little while from the central of the palindromic site but between the same two base of the opposite strand the leaves single stranded portion at the end, and these are overhanging stretch called the sticky ends on each strand, and these are named so because they form hydrogen bond with the complementary cut counterparts. This stickiness of the ends facilitate the action of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecule of DNA, which was composed of DNA from different sources and genomes. So when cut by the same restriction enzyme, the resultant DNA fragment have the same kind of the sticky ends, and these can be joined together end to end using DNA ligase. So here we can see the diagram, the foreign DNA, the vector DNA plasmid, the same restriction enzyme cutting both the foreign DNA and a vector DNA at specific points. Then the ligases join foreign DNA to the plasmid to the recombinant DNA molecule, and then transformation is done. The cell divides in the E. coli, and this diagram is the representing of the recombinant DNA technology. You may have realized that normally unless one cut the vector and the source of the DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. So there is separation and isolation of DNA fragment by using gel electrophoresis, where the DNA is cut by the restriction endonuclease, resulting into fragment of DNA, and these fragment can be separated by the technique known as the gel electrophoresis. Since the DNA fragment are negatively charged molecule, they can be separated by forcing them to move toward the anode under an electric field through a medium or matrix. Nowadays, most commonly used matrix is agarose, which is naturally polymer extract from the seaweed. So the DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it will move. So this separation of DNA fragment can be visualized only after staining the DNA with a compound known as ethidium bromide, followed by exposing the UV light. You cannot see the pure DNA fragment in the visible light and without strand. So you can see bright orange color bands of the DNA in ethidium bromide stain gel exposed to UV light, and this separated band of DNA cut out from the agarose gel and extracted from the gel piece. So this step is known as elution. The DNA fragment are purified in the way as they use the constructing the recombinant DNA by joining them with the cloning vector. So let's know what is a cloning vector. You know that the plasmid and the bacteriophage have ability to replicate within the bacterial cell independent of the control of chromosome DNA. So bacteriophage, because of their high number per cell, have a very high copy number of their genome within the bacterial cell. Some plasmid may have only 1 or 2 copy per cell, whereas other may have 15 to 100 copies per cell, and this number can go even high. If we are able to link a piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or the bacteriophage. Vector use at present are engineered in such a way that they help easy linking of the foreign DNA and selection of recombinant for non-recombinant. PBR322. It is a plasmid, a circular DNA used as a cloning vector in genetic engineering. It helps the scientist to insert and multiply foreign gene inside a bacteria like E. coli. It have some important region. Let's know the main regions of PBR322 plasmid. There is an origin of replication. This is where the plasmid start to copy itself inside the bacterial cell. It ensures the plasmid is replicated and passed on during the cell division. Then ampicillin resistance. It provides the resistance to the antibiotic ampicillin. Bacteria with this plasmid can grow on ampicillin containing media. It is used as a selectable marker. Tetracycline resistance. It provides resistance to tetracycline, and it is also used as a selectable marker. There are specific places which are known as restriction site, where the DNA is cut by using a restriction enzyme. PBR322 have several unique restriction sites inside the ampicillin resistance and tetracycline resistance gene. For example, PST1 cuts inside ampicillin R and BAMH1 cut inside the tetracycline R. Scientists insert foreign DNA at these sites. If a gene is inserted in ampicillin resistance or tetracycline resistance, these resistance genes become inactive, and this helps in identifying which bacteria got the foreign gene. Now, what is the Function of origin of replication. It starts the replication of the plasmid. Ampicillin resistance. Resistance to the ampicillin, and tetracycline is resistance to the tetracycline. Now restriction size allowed the gene to be inserted, and insertion inactivation, it helps in identify the recombinant plasmid. How it is used in gene cloning. A foreign gene is inserted in PBR322 at the restriction site inside the ampicillin resistance or tetracycline resistance. The plasmid is introduced in the bacteria, usually E. coli. The bacteria are grown on antibiotic containing plates, and if the plasmid has the foreign gene inserted in the tetracycline R, the bacteria won't grow on tetracycline. If the plasmid is intact, no gene inserted, and it will grow on both antibiotics, and this method is called insertional inactivation, and it is used to select the correct recombinant bacteria. So it can, the cloning vector have the origin of replication site, selectable marker, cloning site. So origin of replication is a sequence from where the replication start, and any piece of DNA where linked to this sequence can be made to replicate within the host cell. So this sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the targeted DNA, it should be cloned in a vector whose origin support high copy number. Then selectable marker, in addition to ORI, is the vector required to selectable marker, which helps in identifying and eliminating non-transform and selective permitting the growth of the transformants. So transformation is a procedure through which a piece of DNA is introduced in the host bacterium. You will study it later. Normally the gene encoding restriction to the antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin, etc. are considered useful selectable marker for in E. coli. So normally E. coli cell do not carry resistance against any of these antibiotics. Now cloning site, in order to link the alien DNA, the vector need to have very few, preferable single recognition site for the most commonly used restriction enzyme. Presence more than one recognition site within the vector will generally serve fragment, which will complicate the gene cloning. So the ligation of the alien DNA is carried out by the restriction site present in one of the two antibiotic resistant gene. For example, you can ligate a foreign DNA at a BAMH1 site of tetracycline resistance gene in the vector PBR322. The recombinant plasmid will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out for a non-recombinant one by plating the transforms on ampicillin containing medium. So this transforms growing on ampicillin containing media are then transferred on the medium containing tetracycline, and the recombinant will grow in ampicillin containing media but not on the tetracycline. So but now we can know that the ampicillin containing media will have the growth, whereas the tetracycline will not have the growth of the culture. So but the non-recombinant will grow on the media containing both the antibiotics. In this case, one antibiotic resistant gene help in the selecting the transforms, whereas the other antibiotic resistance gene get inactive due to insertion of the alien DNA. So if we insert a DNA, it will become inactive, it will not grow in the tetracycline region, whereas in ampicillin region it will grow. So it helps in selection of the recombination. So selection of recombination due to inactivation of antibiotics is cumbersome procedure because it requires simultaneous plating of one or two plate having different antibiotics. Therefore, alternative selective marker have been used and developed which differentiate recombinants for non-recombinants on the basis of their ability to produce color in presence of chromogenic substance. And this is recombinant DNA is inserted within the coding sequence of the enzyme beta-galactosidase, and this result into inactivation of the gene for synthesizing of the enzyme, which is referred to as insertion of inactivation. The presence of the chromogenic substance give blue color. If there is presence of chromogenic substance, it will give a blue color, and if the plasmid in the bacteria do not have an insert, presence of insert result into the insertional inactivation of the beta-galactosidase gene, and the colonies do not produce any color, and there are identification as a recombinant colonies. Then vector for cloning gene in plants and animal. In this NCERT topic we will know. So we are just going through the chapter. In this we have learned all about the cloning vector and all topics in this. So you may know that, suppose to know that we have learned the lesson of transforming gene into the plant and animal for bacteria and virus, which have known for this ages how to deliver gene to transfer eukaryotic cell and forcing them to do what the bacteria or virus wants. For example, Agrobacterium tumorification, a pathogen of several dicot plant, is able to deliver a piece of DNA known as the tDNA, a transform normal plant cell into a tumor and a direct this tumor cell to produce the chemicals required by the pathogen. Similarly, retrovirus in animal have the ability to transform normal cell into the cancerous cell. A better understanding of this art of delivering gene by pathogenic in eukaryotic host has generally generated knowledge to transfer these tools of pathogen into useful vector for delivering genes of interest to human. So tumor inducing plasmid of Agrobacterium tumorification has now been modified into cloning vector, which is no more pathogenic to the plant but is still able to use the mechanism to deliver gene of interest in a variety of plant. Similarly, retrovirus have also been disarmed and are now used to deliver desirable gene into animal cell. So once a gene or DNA fragment has been ligated into a suitable vector, it can transfer into the bacterial, plant and animal host. Then component host for transformation within the recombinant DNA. DNA is hydrophilic molecule, that is water loving molecule, and it cannot pass through the cell membrane. Why? In order to force bacteria to take up the plasmid, the bacteria cell must be compelled to take DNA, and this is done by treating them with specific concentration of divalent cations such as calcium, which increasing the efficiency with the DNA enters the bacteria through pores and its cell wall. Recombinant DNA can then be forced into such cell by incubating the cell with the recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on the ice, and this enables the bacteria to take up the recombinant DNA. This is not the only way to introduce DNA into a host. In other methods, microinjection, where the recombinant DNA is directly injected in the nucleus of the animal cell. In another method, suitable of plant cell are bombarded with high velocity of microparticle of gold and tungsten coated with DNA is a method of biolistic or gene gun. Now these are important points of this chapter, so note down: microinjection, gene gun, then the last method used is disarm pathogen vector, which when allowed to infect the cell transfer the recombinant DNA into the host. The process of recombination technique, this is very important. I have made a special video on this steps of this isolation, cutting and insertion of this recombinant DNA technology. Do go and watch that video. Then there are other things, for example bioreactants. This is also an important topic. Foreign gene product. So when you insert a piece of DNA in the cloning vector and transfer it into bacterial, plant and animal, the alien DNA get multiplied. Here the DNA is getting multiplied. Almost all recombinant technology has an aim to produce desirable protein. Here there is a need of recombinant DNA to be expressed for foreign gene in the host cell, involving understanding many techniques. After having cloned the gene of interest and having optimized the condition to induce the expression of targeted protein, one has to consider production on a large scale. Now here, what is an recombinant protein? It is important. If any protein encoding gene is expressed in a heterologous host, it is known as recombinant protein. The cell harboring clone gene of interest may be grown on the small scale in laboratory, and this culture may be used for extracting the desired protein and then purifying it by using different separation techniques. Now you must know some of names of these techniques. The cell can also be multiplied in the continuous culture system whereas the use of medium is drained out from one side while fresh medium is added from other to maintain the cell in physiologically most active log and exponential phase. And this type of culturing method produces a large biomass leading to high yield of desired protein. Small volume of culture cannot yield appreciable quality of product. To produce a large quality, the development of bioreactor was made, where large volumes, that is 100 to 1000 liters of culture, can be processed was required, and this bioreactors may be through of a vessel in which raw material are biologically converted into specific product, individual enzyme, etc., using microbial, plant, animal, and human culture. A bioreactor provides the optimum condition for achieving the desired product by providing optimum growth condition. That means it provides the temperature, pH, then salt, vitamins, whatever is required it provide. So here we can know the diagram of this bioreactors, the simple stirred tank bioreactor and staggered stirred tank bioreactor, through which sterile air bubble are there. Then after that, this reactor is usually cylindrical with a curved base to facilitate the mixing of the reactor content. So what it is doing, it is mixing, and the stirrer facilitate even mixing and oxygen availability throughout the bioreactor. Alternative, air can be bubbled through the reaction, and if you look at the figure, you will see that the bioreactor has an agitator system, an oxygen delivering system, and a foam controlling system, a temperature controlling system, a pH controlling system, and a sampling port, so that small volume of the culture can be withdrawn periodically. Then downstream processing, after completion of the biosynthesis stages, the product has to be subjected through a series of processes before it is ready to marketing and finishing product. So this process include separation and purification, which are collectively referred to as downstream processing. The product now has to be formulated with suitable preservation. Such formulation has to undergo through clinical traits as in case of drug, and strict quality control testing for each product is also required. So downstream processing and quality control test vary from product to product. Recombinant DNA technology. It is a technique used to join together DNA molecules from different organisms and insert them in a host organism to produce a new genetic combination. So let's know a step-by-step process. First, isolation of genetic material, that is the DNA. It is extracted from the donor cell using chemicals or enzyme. Then restriction enzyme. It is also known as a molecular scissor. They are used to cut the DNA at specific sequence, and this creates fragments with sticky and blunt ends. Then the next step is amplification the gene of interest. The desired gene is often amplified using PCR before insertion. PCR make millions of copies of specific DNA segment quickly. Then the desired DNA is inserted into a vector like a plasmid or a virus, which carry on the DNA into the host. The enzyme DNA ligase help to seal the DNA fragment into the vector. Then the recombinant vector is introduced in a suitable host cell, often bacteria like E. coli, and this is called transformation. Then not all the cell can take up the recombinant DNA, so the selectable marker like antibiotic resistant gene are used to identify successful transformation. Then the host cell machinery express the inserted gene to produce the desired protein or trait. Then the transformed cell are cloned to multiply to produce large quantity of the desired product like insulin, enzyme, and vaccine, etc. So the application of recombinant DNA technology is used in agriculture, GM crops, BT crops, the golden rice, and in industry, enzyme for detergent and biofuels are used, and in research it is used to study the gene and protein model organism, and in medicines and vaccine like hepatitis B, and gene therapy, and in insulin. So here we are drawing a structure of this T-even phage, that is T4 bacteriophage. This is a complex virus with a head and tail structure. It looks linear lander. or spider under an electron microscope. It is isohedral structure. Here we can see the head. It is also known as the capsid. So this capsid is isohedral with 20 phase polyhedron. It is composed of protein shell capsid made up of capsomere, and it contains a double stranded linear DNA that is approximately 170 kb in T4. So the function is to protect the genetic material and help in DNA injection into the host. Then neck, it is a collar. It connects the head and the tail, act like an flexible joint, and it helps in the transmission of DNA from head to tail. Then tail is a sheet. This tail sheet is a contractile sheet. It is made up of protein. During infection, it contracts to inject the DNA in the host cell. Then the tail core, this inner tube, it lies inside the sheet, and DNA passes through this when injected into the E. coli. Then base plate, hexagonal structure at the bottom of the tail, act like a control center for infection. Then tail fiber, a long fiber, it is 6 leg appendages responsible for host recognition and attachment to the bacterial surface. And then there are pins. These pins are found at the base plate and helps in pricking the bacterial cell. So these pins help in the penetrate the bacterial cell wall. So in T4 bacteriophage, the part of the head, that is the capsid, its function is to store the viral DNA. Then a neck or collar is present. Its role is to transfer DNA from the head to the tail. Then tail sheet, it is a contractile tube, and its function is to contract to inject the DNA into the host. And tail core is a hollow rod. Its function is to for the traveling the DNA through it into the host cell. And there is a base plate which is hexagonal base, and its function is coordinates infection. And the tail fiber are leg-like appendages. They are attached to, which helps in attaching the host surface. And pins or spike are short projections. It help in penetrating the bacterial cell wall. So here's a mnemonic to learn T4 bacteriophage components. For example, the parts head, neck, tail, base plate, fiber and pins. So for that, hungry ninja took big fat pizza. Thanks for watching. Do like, share and subscribe.